Hello, everybody. I want to say thanks for Brunel University because it provides me this opportunity to continue my master and to get my master degree in disease mechanism and therapeutics. And also, I want to thank my fellowship, Kara, which provide an opportunity to bring as an academic student to continue his study due to the war in his country or her country. So I'm so thankful to Brunel and Kara, and also I'm very thankful to all of my tutors, starting with Dr. Bagnarelli, who guided me during all my master trip and until the dissertation step. And I want to I really appreciate and thank Dr. Golden because she guided me properly in my dissertation from the first step to the final step. I want to represent my thankful to her because I guess like I was a kid and she held my hand in every step, do that, do this, and she teach me how to do to think practically and to identify problems and solve them. I want to say thank you for all my supervisors, Dr. Gordon in lab safety, and Dr. Dranis in bioinformatics and cell signaling modules, all my tutors, starting with Dr. Thanos, and I'm very thankful to you all. Today I will present my presentation. It's multicellular protein and their role in the crosstalk between tumor and host cell. There is increase in there has been increase in studies or research about the involvement of multicellular protein in cancer, and this and there is many publication about the role of multicellular protein in the formation of tumor microenvironments, which consist of tumor cells, extracellular matrix, microvessels, and also immune cells. And we know that extracellular matrix is a structural scaffold that help to form the tumor microenvironment because it is a background that help the cells to stuck on it through binding with integrin, and also it's formed as a layer that separates functional cell from the surrounding stroma cell. And also it consists of supporting proteins such as collagen and also proteoglycans that we will know later how they are involved in cancer formation and the creation of a tumor microenvironment. Now, what is the multicellular protein? It is extracellular matrix protein. It's involved in regulation of cell signaling and also involve information of epithelial mesenchymal phenotype that is highly motile cell can escape through blood vessels to reach to the secondary sites where they can colonize, and this will do the metastasis. Also, it's included in a signaling pathway that causes the overexpression of matrix metalloproteinase, which is proteins that involve in the degradation of extracellular matrix through binding with type 4 collagen and cause to degradation of collagen and do the disintegration of extracellular matrix. These multicellular proteins are highly regulated in tissue modeling, such as inflammation and tissue repair, and also it's highly upregulated during the period development, but its expression level is restricted during adult stages. Cancer cells secrete these microcellular protein that go to the surrounding and can create a soil. This soil is suitable for tumor growth and metastasis. Here, the first protein we are interested in, SPARC, which is secreted protein acidic rich in cytosine. This protein mainly it can be extracellular secreted or inside the cell, which is called intracellular, and it can be in the lumen of endoplasmic reticulum, and also it can be found in nucleus. It consists of 3 domains. The first one is acidic domain, which consists of glutamate and aspartate, and it's included in the antibody-antigen interaction. The second domain is follistatin-like domain that's involved in tumor suppression effect of SPARC. So we have to put in mind that SPARC behaves in 2 directions. It can be tumor activator and it can be tumor suppressor through this domain because it suppresses the activin, which is tumor growth factor beta, and also it causes the suppression of vascular endothelial growth factor and inhibit angiogenesis. So it's important to know the function or how SPARC work, what the mechanism that by which the SPARC work, so we can develop later a strategy to target this protein. Also, the last domain here that we have here, this calcium-dependent domain that depends on the calcium concentration in the tumor microenvironment and can bind to different types of collagen, such as collagen type 1, type 3, and type 4, and type 5. And this domain, it causes, in the tumor microenvironment, it causes metastasis because also it degrades the extracellular matrix or cause disintegration. What is the role of SPARC? In physiological condition, I said matricellular protein in general, functional in the tumor, in the tissue remodeling and also in the development. But in cancer condition, these matricellular proteins are highly expressed, and there are many in many literature that confer this matricellular protein, especially SPARC, is highly upregulated, especially in a breast cancer, bone cancer, ovarian cancer, and prostate cancer. And also, it can be downregulated, as I mentioned earlier, through this domain, the follistatin-like domain. It was found in one literature that in a study by in 2010 that this protein, when downregulated, is related to poor prognosis. This is the effect or the function of a SPARC. Mainly, it causes this association or de-adherence of cell and makes the cells separated by removing the coadherin that stuck cell with each other, and also remove the integrin that stuck the cell on the extracellular matrix. So the cell convert from its epithelial type that is stabilized and stuck into mesenchymal type, which is highly motile, that can migrate through the bloodstream and do colonization. SPARC also causes disintegration of extracellular matrix and prevent angiogenesis. Another glycoprotein, which is osteopontin, which also can be secreted or inside the cell, intracellular, in the lumen of endoplasmic reticulum, Golgi apparatus, or inside the nucleus. Also, its function in bone remodeling and homeostasis. But in cancer condition, it's highly upregulated, especially in bone cancer and breast cancer. And it was found that its expression level increased 10 times than normal tissue. And it supports the hallmark of cancer, which is 6 hallmarks of cancer: cell proliferation, anti-apoptotic, has anti-apoptotic property, and also cell motility and evade from immune system. And also it's involved in tumorigenesis and metastasis, because when interact with its integrin receptor, it will trigger many tumor pathway or pathway that involve in tumor, such as phosphatidylindole 3-phosphate kinase and AKT, ERK and MAP kinase, that lead to increase in activation of some proteins such as UPA and matrix metalloproteinase, which involve also in cancer invasion. Here, the last pathway we are interested in, the ERK MAP kinase pathway, is a highly regulated or well-regulated pathway, and it's involved in cell proliferation and proliferation and differentiation. But when this pathway is mutated, it can be involved in cancer. This pathway starts with the interaction between the effector, which is receptor tyrosine kinase, that trigger a complex GRIP2 and SOS. And then when this complex formed, SOS will activate the RAS to convert GDP into GTP. And now RAS signaling pathway will be triggered, that start from RAF, that has kinase activity, that phosphorylate MYC1, and 2 also has kinase activity, and ERK1 and ERK2 that have also kinase activity. When ERK is activated, it will move from the cytoplasmatic side into the nucleus to trigger a canonical pathway, which is the canonical pathway to trigger a protein a gene expression and the protein production. But in our thesis or our project, we are interested in the process that happen in the cytosol, in the cytoplasm, before being go to the nucleus. But these processes is not well understood yet. What the aim of our project? Our working thesis is that MAP kinase ERK1 and ERK2 signaling is involved in regulating of intracellular trafficking and secretion of spark and osteopoietin. Also, to do that, we analyze the transport of endogenous spark and osteopoietin by using immunofluorescence, by using like a microscope, and also with the last system. And we use Western blotting to measurement or to do the evaluate the protein expression level of these proteins. We use two different ways to inhibit spark and osteopoietin by using short-term inhibitor U0126 and siRNA knockdown. I will not speak in details on my methodology, but in question, you are very welcome. We use tissue culture in DMEM medium. Then we use a short-term inhibitor for 1 hour to see the effect of this inhibitor of ERK1 and ERK2 on the trafficking or the secretion of this protein, osteopoietin, and the spark. And also, we use a transfixion by knocking down ERK1 or ERK2 separately, not with each other, to see what their effect on this protein. We studied the images under like a microscope, and we use, as I mentioned earlier, Western blot to see the expression level of protein. Here, the result and discussion. Here, we have Western blot, and we express the bands. Okay, we here the inhibition. The inhibitor result, the first one. We applied the inhibitor on the Ross cell for 1 hour, and we saw that the inhibition of phosphorylation of proteins ERK1 and ERK2 by disappearance of this band. Now we studied the images under a microscope, and we saw that between control and treated, there is differences in localization of osteopoietin. We found that osteopoietin is localized or clustered in perinuclear region before treatment in control, whereas it's evenly distributed in after a treatment. From research, we know that osteopoietin as any protein, when go to Golgi apparatus, it's undergo to post-translational modifications such as glycosylation, sulfation, and phosphorylation. This will increase the molecular weight of a protein to be trapped or to be trapped inside Golgi, okay, or in the perinuclear region. And this result in line with our result that we can see here, the osteopoietin is trapped before a treatment in perinuclear region or in Golgi apparatus region. And this trapping decrease after inhibition, Also, we know that osteopoietin can be found in many isoforms. It can be outside the cell and secreted, or it can be inside the cell intracellular. From the literature, we know that osteopoietin, which is inside the cell, can be found in the perinuclear region and also in the Golgi apparatus, and also it can be found in the perimembrane. And it was found that osteopoietin can bind with actin cytoskeleton, and this will help in the macrophages and osteoblast cell line, and this help in the separation and spreading of this cell. Also, the phosphorylation process of any protein will help to efficient release of the cargo. We know that kinases is very important in the efficient release of the cargo from vessels. Vessels usually like this, and it is covered with cope. And cope contains specific protein. From literature, I have read that some kinase, such as HR, are at 25P. It phosphorylates specific protein on this cope. And this phosphorylation process, it's help in efficient release of the cargo in cis phase of Golgi. Guess if there is any blocker or inhibitor that inhibit this protein, the HRR25P, from doing the phosphorylation process, so the cargo will not release efficiently. It was found in real literature also, ERK1 and ERK2 are both kinases, and both these kinases, if they work in the similar mechanism of the protein kinase I spoke now about. So if we block ERK1 or ERK2 or inhibit them from being work or doing their phosphorylation, they will affect the vesicles from being fused and release its cargo of proteins such as osteoponetin and spark. We counted the cell that contains this clustered localization of osteoponetin before and after treatment, and we found that control cell has more cluster distribution than after inhibition used. And also we compare the cell between the control and the inhibitor that contains the even distribution localization of osteoponetin. And we found in control, we have less cell with even distribution than after using the inhibitor. We studied also the localization, the effect of the inhibitor, the short-term inhibitor, 1 hour, on spark localization. We found both cells before treatment and after treatment express the protein. However, some cells in the control express the protein more. And after the treatment, what we found that the cells express a protein in varying way, and it is different in their the distribution of a spark is different from osteoponetin. It's important to know how spark is work, because spark involved in normal biological condition and also involved in cancer. And the spark is a protein, and there is this protein is engulfed or absorbed or endocytosis by the receptor called stabilin 1. Once the spark is absorbed inside the cell, especially in constitutively activated macrophages, it will undergo the degradation. Also, it was found from literature that a spark is a chaperone, that when secreted from the cell or cancer cell, it goes directly to the extracellular matrix, hold type 1 collagen or bind it, and cause the degradation of this collagen. And from this result, we can conclude that spark play a role as a scavenger chaperone that can shuttle between inside the cell into the extracellular matrix and on the opposite side. We assess the protein expression level against, and we do normalization against GAP-DH, which is a standard protein. And we compare the protein before treatment and after treatment. We didn't find any change in the protein expression level. What we can conclude from this study or this experiment, we conclude that the effect that we saw in using an immunofluorescence images, that inhibitor effect on ERK1 and ERK2 affect the trafficking and secretion pathway of spark and osteoponetin, but doesn't affect the protein level of this matricellular protein. Now the second experiment, which is the transfixion, we use long-term inhibition knockdown here. We use the non-coding control as a control, non-coding siRNA as a control, and we use siRNA against ERK1. Here we knock down ERK1, And here we knock down ERK2. And it appeared by disappearance of the band itself. And we did that separately. Okay? ERK1 alone, knock down, ERK2 alone, knock down. We didn't knock down them with each other. So we got a high efficient of knockdown. Here, what we can see from the graph here, ERK1 protein reduced significantly by 72%, and ERK2 reduced significantly by 76%. Then we studied the images after knockdown experiment. What happened with osteopontin localization? We see here, before treatment, osteopontin localization or trafficking is evenly distributed throughout the cytoplasm, whereas in the application or after ERK1 knockdown, we saw that a protein localized in perinuclear region. Similar result we saw with ERK2. We know that ERK1 and ERK2, both the protein has interchangeable relationship, and they have redundancy between each other. And also, the activity of each protein reflects the protein expression level. When one protein is silenced, the other protein is activated. That means when ERK1 is silenced or knocked down, ERK2 will be activated or phosphorylated. When ERK2 and vice versa, when ERK2 is knocked down, ERK1 will be activated. And we found from literature that ERK1 and ERK2 can compete on the similar substrate. In literature also, we found that there is 284 substrate for ERK2, can ERK1 interact with. So it doesn't matter if we knock ERK1 or we knock ERK2, because we will get the similar result, and this information in line with our result here. So we got the same result if we knock ERK1 or we knock down ERK2. Also, we count the cell that is evenly distributed and clustered osteopontin. We found that in control, we have around 12% clustered cell, whereas in this percent increased to 30% and 35% for ERK1 and ERK2 respectively. After calculation and counting the proportion of cells that contain even distribution of osteopontin, we found the control is around 88%, and this ratio percentage decreased significantly to 24 and 12 and 11 after knockdown for ERK1 and ERK2 significantly. We did the protein expression level to see the effect of knockdown on the expression level of a protein by using Western blot. We didn't find anything affect spark, so the protein expression level is doesn't significantly change. Whereas we found that osteopontin, after long-term application or a long-term knockdown of ERK1 and ERK2, the bands disappear here. This initial result need to a more experiment to confirm it, but we, from reading the literature, we can guess or we can think about that this protein osteopontin, I told you, it consists of many isoforms. And sometimes the low molecular weight protein can be trapped in Golgi, not secreted. So the band disappear here, but this need more study. Conclusion. From literature, we know that matricellular protein spark and osteopontin involved in cancer tumorigenesis and metastasis. And this attracts many researchers to find a way how we can reduce the protein level or the expression level of this protein. But in our findings, we concentrate in using short-term inhibition and knockdown methods to affect the secretion of this protein, not affect the protein level of itself. What we add, our work, what we add in the knowledge, what we add to the literature, what we add more information about matricellular protein. we reached to, or we arrived to a new knowledge of how ERK1 and ERK2 affect the release of matricellular protein from osteoblast, and how cancer cells send signaling via MAP kinase pathway that affects or enhance the secretion of spark and osteopoietin, which may directly influence the tumor metastasis. This will help us to think in future work, so we can track this protein in tumor microenvironment by using nanoparticles that can target this protein and prevent them from being secreted. This is my references. Thank you for your attendance and listening. And any questions are welcome. You are welcome.
Well, very good, Rayam. So is anyone has questions for Rayam? If not, can I ask you something?
Yeah, sure.
I think the effect after the inhibition on the distribution is pretty striking.
Yeah.